that, we're going to transition to gene therapy.
Wow, thank you so much. That picture honestly was taken a couple of years ago, and I should say it looks better than I look right now. So you guys are lucky. Uh, all right. I'm sharing my screen. Can somebody confirm that you guys can see my screen?
Yes, we can see it.
Okay, perfect. All right. So it says I'm going to be talking about history of gene therapy, but let me tell you that we are living the history right now. So rather than talking about the history, I'll talk about the current state of gene therapy for sickle cell disease. These are my conflicts of interest and disclosures. I work with a lot of different companies that are developing gene therapy clinical trials, and I work with them to improve how we develop these therapies. But none of the work that I'm presenting today is related to any of these disclosures. So I'll briefly start with the genetic basis of sickle cell disease and then compare how bone marrow transplant that Dr. Ivy was talking about compares to gene therapy. We'll then dive into the different types of gene therapy and talk about some of the results of clinical trials. And then towards the end, we'll talk about how do you pick which treatment you should choose. And so I show this slide to most people that I'm talking about. In order to understand a genetic disease, you have to understand where genes are. So, as you probably know, we all have trillions and trillions of cells in our body. And inside those cells, we have chromosomes. And on these chromosomes, we have genes which are made up of DNA. So, in order to compare this thing to something we are more familiar with, if you think of cells looking like books, the chromosomes are essentially like book chapters, and the sentences in those chapters are the genes. And just like you can have a spelling mistake in a sentence, you can have spelling mistakes in these genes. And sickle cell disease is nothing but a spelling mistake in the gene that codes for hemoglobin molecules. And so on chromosome number 11, inside blood cells, we have a gene which codes for the hemoglobin molecule HBB, and instead of GAG, there is a GTGA, and that's the spelling mistake. As Dr. Ivy also mentioned, there are many, many treatments available for sickle cell disease right now. We have known about blood transfusion since the 1980s. Hydroxyurea has been around for many, many years. And within the last 10 years, L-glutamine, crizanlizumab, and voxelotor were also FDA approved. But the issue with all these medicines are that they need to be taken on a regular basis, either every day, every week, every month, or even if you take them regularly, they may not work for you, and they don't work the same for every individual across their lifespan. So they may work for you initially, but then they may not work for you later on. So it's a very different sort of approach with each one of these medicines, which is why we don't think that they are a curative therapy. We've also known about bone marrow transplant, or BMT, since 1983, when it was first performed. And then last year in December, not one but two gene therapies were FDA approved for sickle cell disease. And so, how do we do bone marrow transplant? Let's talk about that first, and then we'll dive into gene therapy. So, let's say this is an individual with sickle cell disease here, and on the right side, this is what their blood looks like. As you can see, there are a number of sickle cells over here. So, the first thing that we have to do a transplant is to find a donor. Now, the best donor is usually a brother or a sister who's a full match to you, but does not have sickle cell disease. Now, the chances of finding a brother or sister who's a full match but does not have sickle cell disease is usually 1 in 5 to 1 in 10. So imagine if you have 5 to 10 siblings, maybe one of them will be a full match and doesn't have sickle cell disease. So that's a fairly rare chance, if you ask me. Now, for those lucky individuals that have a fully matched donor, we collect stem cells, bone marrow stem cells from that donor. Then we give the patient with sickle cell disease chemotherapy and or radiation. And the purpose of this chemotherapy and or radiation is to kill the blood cells that make sickle cells. So after chemotherapy, their bone marrow becomes empty. And then we take these cells that we have harvested from the donor, and we put them back into the patient and wait for a couple of weeks. And as these blood cells start growing and start making new blood cells which look just like the donor, our patient does not have sickle cell disease anymore either. Now, that all sounds very simple, But of course, there are problems with this. First thing, as I mentioned, not everybody has a donor available. But even if you have a donor available, there are some immune problems associated with bone marrow transplantation. The two main being graft versus host disease and graft rejection. Whenever we put cells from one person into another person, there is a chance that the recipient may reject those cells, which causes graft rejection, or the cells go into the patient and start attacking the patient, something called graft versus host disease. And both of these problems can be life-threatening if they're not controlled properly. When we use chemotherapy, people lose their hair. This is temporary, but it's a big deal for many individuals. Also, chemotherapy exposure can affect the way your kidney, your ovaries, and testicles work. And so some individuals may not be able to have children after receiving chemotherapy. And then chemotherapy also reduces your ability to fight infections, at least transiently. So there is a risk of catching infections, and there is a low but non-zero risk of developing cancer and maybe even dying because of the exposure to chemotherapy. Now, while we can't fix all of these problems, the first three problems over here, not having donors or the risk of graft versus host disease and graft rejection, we can address that if we use a person's own cells. And so that's where gene therapy comes in. So in gene therapy, once again, this is a person with sickle cell disease, and on the right side, this is what their blood looks like. You can see there are a lot of sickle cells. In gene therapy, instead of finding a donor, we actually collect stem cells from the patient himself or herself. We then take these cells to the laboratory where we use either a lentiviral vector or CRISPR-Cas9 protein to genetically modify these cells. Once these cells have been modified, we still have to use chemotherapy to wipe out the sickle producing cells. And once these cells have been completely wiped out from the bone marrow, we infuse these genetically modified cells into the patient and wait for a couple of weeks for them to grow. And as they grow, they start making normal blood cells, and the person doesn't have sickle cell disease anymore. Now, there are many different kinds of gene therapies. We can do gene addition, gene replacement, we can increase fetal hemoglobin by a number of different methods. And last but not the least, we can fix the sickle cell mutation. Now, that last part over there, fixing the sickle cell mutation, it's much harder to do than it is to say. And so as of today, there are very few efforts or clinical trials which are actually fixing the sickle mutation. There's actually one clinical trial going on in California, but the first two approaches of gene addition and increasing fetal hemoglobin, these are the ones which are FDA approved. So let's talk about gene replacement or gene addition first. Now, as I told you, sickle cell disease is caused due to a spelling mistake, right? And so here I have a sentence which has two spelling mistakes, as you can see. What this technique does is it basically adds the normal spelling of the gene somewhere in the sentence. So in your genes, they will add a normal copy of the gene, which the normal spelling is, somewhere. And we use a modified HIV virus to do that. That's a lentiviral vector. Now, this is a HIV virus from which the HIV causing machinery has been removed. So this virus cannot cause HIV/AIDS, but these viruses are very good at transferring genes inside blood cells. So we use them not just for sickle cell disease gene therapy, but for many, many gene therapies. And it's very safe and it does not cause HIV. So we use that to put the normal spelling of the genes somewhere in the sentence. Um, and this is the method. So what we do is we take a empty HIV virus, a lentiviral vector, we package a normal gene inside, and then this virus goes inside the blood forming cells, delivers the normal gene, and now you make normal hemoglobin again. So this method is what was used to develop this product called Lovotibegdogene autotemcel or Lovocell. The trade name for this is Lyfgenia, and this is an FDA-approved medicine for individuals who are 12 years and above. This was developed by a company called Bluebird Bio, and on their clinical trials, they treated at least 47 patients who were followed for about 1 month to 60 months. And as you can see on the right-hand side, these are the average hemoglobins of the individuals who were treated. And before infusion at baseline, the average hemoglobin was about 8, but after treatment with this, the hemoglobin rose to about 10 to 12, around 12 grams, which is near normal. And as you can see in the blue over here, half of that hemoglobin was coming from the virus that was introduced. So by doing this individuals, instead of having sickle cell disease, they become like somebody who has sickle cell trait. The next approach is to increase fetal hemoglobin. Now, we've known for a very long time that if you have more sickle hemoglobin, you have more pain, more acute chest, poor quality of life, and a shortened lifespan. Hydroxyurea increases fetal hemoglobin, and that reduces pain, that reduces acute chest, improves quality of life, and improves lifespan. Now, if we could do that, if we could raise fetal hemoglobin without having to take hydroxyurea every day, that would be fantastic. And that's what gene therapy does. So once again, here is the spelling mistake. Now, we can use CRISPR-Cas9, which are like these small molecular scissors, which go and cut the genes at any place that we want them to. So by cutting the genes, we can introduce random words within those sentences. And so this is essentially what it looks like. In scientific language, these are called insertions and deletions, or indels. So sometimes when I'm describing this technique, instead of calling it gene editing, I like to call it gene vandalism. But I can tell you that the gene editors don't like that very much. So let's just stick to gene editing right now, but it's gene vandalism essentially. But what happens is this sort of gene vandalism is good for us because by doing this, we can raise fetal hemoglobin. So this was developed in a product called hexagamglogene autotemcel. The trade name is Casgevy. This is also FDA approved for individuals over 12 years. And it was developed by two companies, CRISPR Therapeutics and Vertex Pharmaceuticals. On their trial, they treated 44 patients and followed them for about 1 month to 5 years. And again, as you can see on the graph at the bottom, the average hemoglobin went up from about 8 grams per deciliter to about 12 grams per deciliter in these individuals, and at least half of that was coming from fetal hemoglobin. And so once again, these individuals, because they have so much fetal hemoglobin, they don't have sickle cell related crisis after that. And most of the individuals on these two trials have not had any sickle cell related pain crisis after undergoing gene therapy. There is another product which is currently in development by a company called Editas. This is called Rainy-Cell. This is not FDA approved right now. This also uses a similar CRISPR approach to raise fetal hemoglobin. They have treated 11 patients so far. And once again, the results are very similar to the previous trials that we have seen. So, as I mentioned earlier, CRISPR-Cas9 does this. It changes, it adds random words or alphabets within the sentences. It would be ideal if we could go and correct the spellings. And base editors, which are even a more newer technology, can do that. But this is yet to come in clinical trials. Now, I told you earlier that there are some problems with bone marrow transplant and how gene therapy can fix at least the first three problems because every patient can potentially serve as their own donor. You don't have to find a donor. But the problems listed here on the bottom still remain. Most clinical trials and most products, gene therapy, still require you to take high dose chemotherapy. And these chemotherapy can cause hair loss, infertility, infection, cancer, death. All those things can still happen. But more than that, we run the risk of having some unknown risks. And so a couple of years ago in 2021, Bluebird Bio announced that two patients on their clinical trial developed leukemia. Now, um, viruses can also cause leukemia. And so if you have, this is what our lentiviral virus is supposed to do. It is supposed to insert the normal spelling at the end of a sentence. But if it inserts the normal spelling at the wrong place in between a sentence, it could even further change the meaning of that. And this is what happens if when we have a virus that causes leukemia. We call it insertional leukemogenesis. Now, this is not what happened on the Bluebird trial, though this is a theoretical risk. It could happen. Extensive evaluation was done, and these two papers were published subsequently, and they clearly showed that the virus was not involved in the development of leukemia in these patients. But nevertheless, these patients got high dose chemotherapy, and we know that chemotherapy exposure can also cause leukemia. So we wonder whether that might be what happened. Now, since then, since those two patients, no other patient has developed leukemia, but that risk is still there. And we need to talk about that. And we need long-term follow-up from these studies. These studies have only followed patients for about 5, 6 years. We need to follow these patients really, really long, 15 years, 20 years, to really understand what the risk is from these novel therapies. I also want to bring up this question of what a cure means. It probably means different things to different people. You know, some would say if they don't have any sickle cell associated pain crisis, or they don't need medications or blood transfusions, that's a cure. But what I like to tell patients is if your body is like a wall, sickle cell is disease is like a hammer which keeps chipping on that wall every single day. Transplant and gene therapy takes the hammer away, but it doesn't really repair the wall. So the damage that's happened to your body because of sickle cell disease before you undergo gene therapy or transplant, like if you have had a stroke or osteonecrosis or heart damage, kidney damage, lung damage, that will remain and will continue, and you will still have to see a physician after undergoing gene therapy or transplant to take care of those. Gene therapy and transplant only affect blood cells. So if an individual with sickle cell disease were to have children, they can still pass on the sickle gene to future generations. And so we need to make individual assessments for every patient, whether gene therapy or transplant is right for them. Remember, even on the gene therapy clinical trials, there are some patients who continue to have pain afterwards. There are very few, a handful of them, but they are not zero. There are some patients who can continue to have pain after undergoing gene therapy. So, once again, cure is a very heavy word, and what it means for different people will need to be individualized for them. So what I would leave you by saying is that gene therapy is surely transformative, hopefully in a good way, but it could cause some bad things too. And so this is the question I am usually asked. Which one of these therapies should I pick? Which one is the best? Just tell me and I'll do that. The short answer is we don't know. I don't know. I can't tell you if one of these technologies is better than the other. Surely there are very impressive short-term results, 5 years, 6 years, but we don't know what the long-term safety and efficacy of these therapies is going to look like, whether one is going to be better than the other. So what I usually tell patients is if there is a trial available and you have severe disease, consider participating in that trial. It will probably help you, but at least it'll help advance these therapies for all other patients around you. And St. Jude does have a gene therapy trial, which is open and enrolling. So if you are interested, please contact me and surely discuss with your family and doctors to see if this is the right option for you. There are known risks and there are unknown risks, and it's a decision that is highly personal, and you should all make that decision for yourself in discussion with your physician and your family members. And so this is that shameless plug that I was telling you in the last slide. That we have a clinical trial at St. Jude, which is currently enrolling individuals between 18 to 25 years. If you have SS, S beta 0 or S beta plus disease and have severe disease, which is defined as having at least two sickle cell related events per year for the last 2 years, please reach out to me. My email address is here at the bottom, akshay.sharma@stjude.org, and I'll be happy to discuss this further with you. So with that, I thank you very much for your attention. And this is one of my patients who gave me the permission to share his picture over here. He and I partnered together, and he decided to participate in a clinical trial a couple of years ago, and he's about 3 years out and does not have sickle cell disease anymore. And I hope that all of you listening in the audience can have a similar fate as he did. So thank you very much once again for your attention, and I'll be happy to take any questions.